After the exciting talks we had before on our animal model and on our observation from the clinical work, I would now like to show you some data on the stabilization of viral vectors in the CMC area of drug product development. And we at Leukocare use an algorithm-based development approach with which we can yield a good stabilization of viral vectors and viral vaccines. The presentation will give an intro, then show two different projects, a stable liquid adenovirus formulation, as well as a dried influenza vaccine work. And after some conclusions, I would also show you references and the contributions to the work. Leukocare is a formulation development company located in Munich in Germany, as well as now since this year also in Milford, MA in the US. We have about 60 employees with lab and offices in both locations. Our formulation development approach starts with a thorough understanding of the drug substance and a full design space for excipients. And then we employ our database that comprises a lot of experience on and information on different excipients and their combinations to stabilize proteins as well as viruses. And we use algorithm-based formulation selection to work with this database, and a software-based evaluation by linear as well as nonlinear statistics helps us to continuously narrow down the design space of potential excipients and excipient combinations until in the last step we do a final optimization and then show the stability of the new developed formulations in a 3-month stability study. The motivation for this work is that a rapidly growing importance can be seen of recombinant viral vectors, adenovirus as well as adeno-associated virus, and vaccine development as well as gene therapy. Still, the viral vectors are prone to degradation due to stress by manufacturing, storage, and distribution. So stability issues are a substantial hurdle for development and administration of viral vector-based vaccines and gene therapeutics. And insufficient drug substance stability impacts product quality, efficacy, and safety, and can thus reduce product potential and increase costs. And we think that formulation development offers a route to stabilization of virus. Here is the first project I would like to present today for a stable liquid adenovirus serotype 5 formulation. The pre-selection of excipient was done from our technology platform comprehensive database that employs about 100 different excipients, and are typically listed in the pharmacopoeias and are well known. We did here a DOE approach. The design of experiment formulation matrix was built for the adenovirus serotype 5 on a pH of 7.4 with sucrose and magnesium chloride as a basis in the formulation buffer. And amino acids were used as variable excipients, whereby the amino acid itself was considered as a factor of the DOE. You can see here's a list of amino acids we tested. And the concentration levels were used as the levels of the DOE matrix. The D-optimal design was chosen to calculate the different compositions, and we resulted in 40 different formulations. Those were stored at 3 different temperatures in a storage matrix, so at 37 degrees, 25 degrees, and 5 degrees for different storage durations. The results were done by hexane staining to test on the infectivity units in the different samples. And you can see here on the x-axis all the different formulations listed. We have the infectivity units here on the y-axis, and each bullet here represents one testing results at 37 degrees Celsius. Blue is for the beginning, and then the red ones are the 14-day storage results, green for the 21 and purple for the 35 degrees. You can easily recognize it's just one bullet here for the long storage of 35 degrees at 37 degrees Celsius. That is formulation F129. That also has the best result for the 21-day storage. However, in total, the quite varied results help us a lot in DOE evaluation to assess the stabilizing amino acids, and so that do the job not that well. On the right-hand side here, you can easily recognize that all three plots look rather the same. And this is exactly what we want to show, that stability at 37 degrees can be predictable for storage at 25 or 5 degrees. And we could see here for all of these different storage temperatures, a very good stabilization by methionine and a less stable stabilizing potential by tryptophan. However, when we look deeper into the statistics and applied multiple regression modeling with the least square model, what we could see also that especially on the 5-degree data, what we found is that the negative impact of the tryptophan combined with the positive result of the methionine. So if you would look on the interaction of the both, you then saw a very positive stabilizing effect. So in addition to the simple linear regression for DOE, if we did also the interaction analysis, we found that interaction of excipients can have a tremendous positive effect that would not be expected from the linear regression. We then optimize the formulations in a one-factor time or OFAT approach, focusing on methionine, alanine, and glutamic acid. We mainly optimized this formulation I pointed out before, as well as the second one. We eliminated tryptophan and magnesium chloride, replaced sucrose by mannitol in a test, also tested for lysine, again with pH 7.4 and an osmolality of about 450 to 650 milliosmol per kg. The results are plotted here, and we combined the stability results from the first DOE round, you saw data before, as well as from the optimization round here. What we can easily see here is for the 37 degree stability data, that after 14 days of storage here, we have about 1 log loss and about 2 log loss for the 21 degree storage in the beta stabilizing formulations. And if we go then here to the optimization round, we saw that we have at 21 days almost less than 1%, 1 log loss. So a tremendous optimization. And we see here the purple line looks pretty well. That is this formulation F24, which also turns out to be the best for storage at 25 degrees storage, the only one that yielded detectable results even after 12-month storage at 25 degrees. Whereas at 5 degrees, they look more or less the same. We had the same picture here, but still you might remember that I showed you before with nonlinear, with multiple regression analysis, we could detect excipient effects even in a bunch of formulations that more or less looked all the same. So as a summary, the design of experiment approach together with accelerated storage at 37 degree is predictive for real-time storage and can use to shorten development timelines, specific amino acids identified with positive or negative effects on stabilization. And we think that a well-balanced combination of components is important regarding concentration and stabilizing interactions. And for further information, I put the citation here. The second project I wanted to show you was a project to find solutions to overcome barriers in viral vaccine manufacture, we often notice one is that aseptic manufacturer is quite taking a lot of efforts, especially during fill and finish, and we thought that terminal sterilization might be an option here, and also to have room temperature possible to avoid cold chain. And our idea was to start with a spray-dried formulation with subsequent terminal sterilization, and the dried form could allow for higher storage during and transport temperature, the spray drying as an alternative to capacity limited lyophilization, and protective formulations that would at all enable the terminal sterilization as well as the spray drying, which is not a typical thing to do with virus. The concept was to use a pandemic's influenza vaccine from GlaxoSmithKline. And we only use the virus part of it, not the adjuvant. It's a liquid dosage form for injection to which the adjuvant is later on added. We did a reformulation by buffer exchange by dialysis and formulation development, a DOE design of experiments as well as an OFAT approach again. The processing comprised a spray drying of the rebuffered material with subsequent terminal irradiation, and proof of concept was shown in vitro by different analytical methods, so size exclusion, HPLC, SDS page, dynamic light scattering, and potency with the hemagglutination assay. And we also did an in vivo proof of concept by a vaccination study in non-human primates with the readout of hemagglutination inhibition assay and microneutralization assay. So here the DOE formulation matrix, again, a fractional design was used. We tested as factors, amino acids, sugars, and the levels were both two concentrations here. And it turned out that trehalose as a sugar with the highest concentration in amino acids, as well as the sugar, worked best. And we also added two further stabilizers for the animal trial. And spray drying and assessment of the spray dried material was done by the group of Professor Shirleys in University of Kiel in Germany. The in vitro results show by dynamic light scattering that at 5 degrees Celsius, there is hardly a change to be seen. So here in the back is the beginning, is the 0 value, and then we have 1, 3, and 6 months of storage results. However, at 25 degrees storage of the trehalose stabilized material, we see here that the front line, the yellow line, started to shift to the right, meaning we have a higher diameter of the virus, which is a typical indication of a beginning morphological change. So here we would say that stability can be assigned for 3 months at 25 degrees, but not for 6 months. On the size exclusion chromatography side, we have plotted here all the different formulations in the beginning of stability storage. So in front here, you see the red one, and I just follow that line. We have some minor components, one major component, again, a minor component. Here's a second major component, and here it runs out. We see that if we have a spray drying with just mannitol, we keep the minor components, but they are changed. And if we add then terminal sterilization, all is lost. That is a blue form here in the second part. With the trehalose as a stabilizer, also we lose the minor components, as you can see here. At least the major components are fully maintained. That what we can also see during the stability storage here in front is the starting value, in the end then the 6 months at 25 degree value. And we can see that it all looks the same. So although the minor components are lost during irradiation, we have a full stability then during the storage at 25 degrees for 6 months, which was the maximum time we tested for. Here now the results from the in vivo potency testing that were done by Public Health England. The test system were macaques, Macaca fascicularis, mature adults with an equal distribution of males and females, n equals 6 per group. However, the negative control group with PBS, there were only 2 animals. The test articles we used was a negative as well as a positive control. So the negative control here in the beginning, the PBS, the positive is the original Pandemrix. And then we had 3 different spray-dried material test articles. It was the mannitol stabilized formulation after terminal sterilization. We had the trehalose stabilized material without gamma irradiation, and we had the trehalose stabilized material after gamma irradiation as a comparison. And what we could see here in the hemagglutination inhibition assay, as well as in the microneutralization assay, is that we have almost same result here prior to vaccination and after vaccination. We see that the negative control is maintained. We just see a slight increase here. Positive control exerts its activity, and we can see that here the trehalose stabilized materials are more or less the same as a positive control. There's no significant difference. The green bar represents the mannitol stabilized material, and here we have a significant lesser effect, and results for both tests are almost identical. So the outcome here was that the immune response in non-human primates was fully retained after spray drying and irradiation using a formulation that was terminally sterilized compared to a non-irradiated original Pandemrix. The storage at ambient conditions, 25 degrees and 60% relative humidity, did not show an influence on the antigen integrity and activity. And these results are also summarized in a paper that is referenced here. So in conclusion, we could see that highly effective pre-selection strategy of stabilizing excipients by an algorithm-based development approach, together with an accelerated aging model, offers the opportunity to develop a stable liquid formulation of viral vectors or viral vaccines in a short development time. And in addition, for production of dry vaccines, a process of spray drying followed by terminal sterilization also offers a potential alternative to lyophilization. And especially the adenovirus, you might already know, are also very often used for the COVID-19 vaccines. And what we showed here for the adenovirus stabilization might also help to find ways to have easier storage of these materials. So in the end, I would conclude my talk with the references and contributions. The first project on the stability of formulation development of a stable liquid 85 formulation was a Leukocare internal project, and the optimization of the split virus formulation for spray drying was a collaboration project with the University of Kiel and Public Health England. It was supported by the German Bundesministerium für Wirtschaft, so it was publicly funded, and the Pandemrix was kindly provided by the government of Brandenburg in Germany. I would like to thank you for your attention, and please feel free to address any questions you might have. Thank you.